Hello students, today it is our new unit, that is unit 7, genetics and evolution. Genetics and evolution. Now here first question is, what is genetics? Genetics. And who used this term genetic first? The genetic term was used by Bateson, right? First, this genetic term is used by Bateson, right? So what is genetics? Genetics means it is a collective study of heredity and variation. Genetics means a collective study of heredity and variation. Okay. And evolution means what? A gradual variation comes, right? Means when a differentiation comes, a gradual differentiation happens time after time, or we can say due to the variation, right? A gradual variation happens, and this is called evolution, right? In this unit, two things are there. We have to read that genetics, another one is our evolution. And so here only 3 chapters are also there. That is, first one is our principle of inheritance and variations. Principle of inheritance and variations means here we read about the Mendel's law of inheritance, right? Other law of inheritance also there. Chapter 5th, it is molecular base of inheritance, right? Means DNA or genes, or we can say DNA fingerprinting or human genome project. We will read this one, right? At molecular base. And chapter 6 means it is evolution, right? How we can say how this modern world is evolved, right? How today's all the animals and plants which are present in our earth, how they evolve, right? So we start from there. Here only we need to read these statements, because in NEET exam we know some statement come from this one also, right? The work of Mendel and other who followed him. Work of Mendel and other who followed him. The other means what? Like that is our Hugo de Vries, Karl Correns, right? And others who followed him gave us an idea of inheritance pattern. However, the nature of those factor means here, because Mendel used this factor term, right? At that time he has no idea, or at that time no one have idea about the genes, about the DNA, right? Which determine the phenotype was not very clear. Which determine the phenotype? Phenotype means what? Now one word is coming here, phenotype. Okay, I will tell you about all the terms which is used in genetics, right? Phenotype means what? Physical appearance, right? Morphological appearance. That is called phenotype. And genotype means it is the genetic constitution, right? We can say how a gene is formed. That is called genotype. As for example, capital T is small t, it represents the genotype. But phenotype, what is? If capital T is present, this tallness is the phenotype, which we can see, right? As these factors represent the genetic basis of inheritance, understanding the structure of genetic material. What is genetic material? Means DNA material, and the structure basis of genotype and phenotype. Already I tell you, genotype means that is which type of genes are present here, right? And this is the constitution of a gene or a factor. And phenotype means what? Physical appearance. Conversion becomes the focus of the attention in biology for the next century. The entire body of the molecular biology was consequent development with major contribution from Watson, Crick, Nirenberg, Khurana. These are the scientists, right? Who has the major contribution in this field? Watson, Crick, Nirenberg, Khurana, Korenberg. That is father and son. Nirenberg and Korenberg. Banger, Monod, Brenner, extra. A parallel problem being tackled was the mechanism of evolution. How the variation comes and how a new species is formed. Right? Here, a awareness in the areas of molecular genetics, structural biology, and bioinformatics have enriched our understanding of the molecular basis of inheritance or evolution. Right? What is given here? Awareness in the area of first molecular genetics, that is genes, DNA, RNA, expression of genes, structural biology, that is we can say morphology and anatomy, and bioinformatics, systematics have enriched our understanding of the molecular base of evolution. In this unit, the structure and the function of DNA and the story and the theory of evolution have been examined and explained. In the next chapter, we will read this one. Now, two scientists are given here, Watson and Crick. Watson and Crick, who first give us the idea about the structure of DNA, BDNA, right? And so, we can say here, Watson and Crick model of DNA. James Davy Watson was born in Chicago on 6th April 1928. In 1947, he received BSc degree. It is not very important for us, right? Okay, you read this one, right? Okay, during this year, his interest in bird watching has matured into a serious desire to learn genetics. This became possible when he received a fellowship for graduate study in zoology at Indiana University, Bloomington, where he received his PhD degree in 1950 on a study of effect of hard X-ray and bacteriophage multiplication. You know bacteriophage, right? Virus. He met Crick and discovered their common interest in solving the DNA structure. At that time, we have different because at that time no specific DNA structure is given here, right? Their first serious effort was unsatisfactory. Their second effort, based upon more experimental evidences and better appreciation of the nucleic acid literature, resulted early in March 1953 in the proposal of the complementary double helical configuration. Double helical configuration of DNA molecule, it is proposed by when does early March in 1953, right? Francis Harry Campton Crick was born on 8 June 1916 at Northampton, England. He studied physics at University College London and obtained a BSc in degree. In 1937, he completed PhD. In 1954, on a thesis that is entitled X-ray diffraction, polypeptides and proteins. You know polypeptide means what? That is a protein. A critical influence in Crick's career was his friendship with J.D. Watson. That is, the critical influence in Crick. Sometimes what happens in our life, we understand. That is, sometimes we say them connecting dots. Connecting dots means sometimes what happens, you will meet a person who will change your life, and that's happened here also. This terms is philosophical. It is called as connecting dots. Your future is totally depend upon the connecting dots. Simply, you meet some person, and so your life, total line is changed. Life of line is changed, right? That's here. The critical influence in Crick careers was his friendship with J.D. Watson, then a young man of 23, leading in 1953 to the proposal of the double helical structure for DNA and the replication scheme. Crick was made an FRS in 1959. The honors to Watson and Crick with Crick include the John Collins Warren Prize. Who will? Watson, Crick, John Collins also here. Warren Prize of the Massachusetts General Hospital in 1959. The Lasker Award in 1960, the Research Corporation Prize in 1962, and above all, the Nobel Prize they got in 1962. Watson and Crick. Do you understand? Okay, now for first this. In this unit, this is the first chapter, or we can say in 12th section, it is the fourth chapter. Principle and inheritance of variation. In this chapter, we have 6 points to read. Mendel law of inheritance, inheritance of one gene, inheritance of two gene. Inheritance of one gene means monohybrid cross, inheritance of two gene means dihybrid cross, inheritance of three gene means trihybrid cross, tetrahybrid cross, or we can say polyhybrid cross. Sex determination, male or female, mutation, right, sudden changes, and genetic disorders. Means chromosomal disorders, aberration disorders. So before going to start this chapter, I think I have to tell you some important terms which are used, right, which are used in this chapter. Okay. First, this one, genetics. This term was first used by Bateson. Bateson, right? First thing is there. Full name is W. Bateson. Okay, Bateson. Second, what is genetics? Genetics means genetics. It is the actual genetics means what? We can say here heredity plus variation. We collective study of heredity and variation. Heredity and variation. That is genetics. The question is, what is heredity? That what is heredity and what is variation? Heredity means what? You can say heredity. Heredity means we can say transmission of genetic characters from parent to offspring. Transmission of transfer of genetic traits from parent to offsprings. That is called heredity. Now, what is variation? Second term is heredity and variation. Variation means variety, right? Means different. Individual have some differences. Differences means individuals, they have differences from their parents. These differences, because they are different from parents, these differences are called variation, right? Differences in offspring or individual, we can say also. Individuals is called as variation, right? That is, so these are some terms. What is genetics? Who use this term? What is heredity and what is variation, right? Now here other some important points which is not in our NCERT, right? But these names are present in our NCERT, so we have to read this. Who is the father of genetics? First question is, who is the father of genetics? Father of genetics. Who is the father of genetics? Simply we can say here Gregor John Mendel. Who is this? Is Gregor John Mendel, or shortcut I write here Gregor John Mendel, right? Second point is our who is the father of genetics? Second is who is the father of modern genetics? Father of modern genetics. Who is the father of modern genetics? Modern means when we come to molecular basis, it is Bateson, that W. Bateson. Both are same, right? Now, third portion is coming here, experimental genetics. Who is the father of experimental genetics? Sometimes it is also comes. Father of experimental genetics. He was Morgan, because all these experiments means all these thesis of this which one? Morgan. He used Drosophila, fruit fly, and also gave many terms, right? So that is Morgan. He was called the father of simply Morgan. We will read the contribution of Morgan in the further place, right, in this chapter also, right, because he experiment which animal he used here, Drosophila, commonly called as fruit fly. This animal he used, it is scientific name, so we underline this, right. He used the term linkage, crossing over, right. Morgan used these terms, linkage, crossing over, crisscross inheritance, right, and the terms also there, sex linkage. And last one is there, who is the father of human genetics and biochemical genetics, right, we can say last point, father of human genetics and biochemical genetics. Biochemical genetics, right. So he is A. Garrod. A. Garrod. He is called because he first observed the genetic disorder, that is alkaptonuria, or we can say here black urine disease, alkaptonuria, first discovered. And he also give a concept, then one his concept is, he give the concept, which concept, one mutant gene means what, one metabolic block. This concept he gives us. One mutant gene, if one mutation is occurs, that means that metabolic is blocked, a new metabolic activity is going on, right. So these are some, this is means, this one we can say here, it is the history or the father of the genetics, father of modern genetics, father of experimental genetics, or father of human genetics and biochemical genetics, right. And I think you know, father of genetics means Gregory John Mendel, father of modern genetics he is W. Bateson, father of experimental genetics it is that is Morgan, and he used which animal, Drosophila, right, fruit fly. And father of human genetics and biochemical genetics is A. Garrod, right. First he observed the genetic disorder, that is our alkaptonuria, and he also gave a concept, that is the one mutant gene means one metabolic block, right. Some other genetic terms which is used in our genetics, these are, okay, now we move here, terms used in genetics, right. So here first, terms which are used in genetics mostly, right. So first, factor, because factor I write here factor, actually factor, this time factor is not used, right, but Mendel, at the time of Mendel, at that time we have no idea about the genes, at that time they had no idea about the genes, and Mendel used this term factor, right. So factor means according to Mendel, who is this factor, Mendel. So according to Mendel, this factor means what, this factor is the unit of heredity. Mendel, according to Mendel, factor means what, unit of heredity. Unit of heredity, right, this factor. And factor what is used by Mendel's, okay. Mendel only not used this factor only, he also used elements, but Johnson, when Johnson come, right, so Johnson, this factor, then gene, I write here gene, then we understand. Genes means who comes, Johnson's, right, right? This Johansson's, he used the term genes in place of factor, right? Johansson used this term gene, and now gene is the unit of heredity. What is gene? Gene means what? It is the unit of heredity, right? And Morgan what? Morgan used the means capital letter and small letter for the dominant and recessive genes. Okay, you understand this? We can say here one important point is there, so I write here Morgan used symbols for factor here, or we can say here gene, right? Dominant for dominant capital letters and for recessive he used small letters, right? Capital letter and small letter. As for example, like capital T, small t. Morgan used. Now allele, another term is our third, allele. Allele, full form of allele means allelomorph, right? That means what? Allele means what? The alternatives of genes which are located on homologous chromosome on the same loci or on identical loci. Loci means what here? Locations, right? You can see alternative forms located on homologous, located on same loci. Loci means what? Location of homologous chromosome. Alternative form of gene, we can say form of gene or factor. Okay. After allele means here homologous. I use here word homologous. Homologous or homozygous. Homo means same, right? Same pair. We can say same pair, within the same pair. Now another one is homozygous. Homozygous. Z. This one is Z, right? Homozygous means what? Look here. If genetic constitution is same or identical, then it is called homozygous. If genetic constitution is different, right, then it is called as heterozygous. Homozygous and heterozygous, right? Homozygous means if genetic constituents is different. That means what? It contains, this is not same, it is not different, it is identical. Is same. As for example, capital T or small t, right? And heterozygous means what? Heterozygous means genetic constitution is different. Means alternative form. As for example here, capital T, small t. That is called heterozygous. Okay. Phenotype means Another word is phenotype. Phenotype means physical appearance, right? How they looks, what we see, right? Here we can say external or morphological appearance, right? This tallness, as for example, we can say here tallness, right? And or dwarfness. And genotype means what? This genotype, actually, it is this one is the constitution of genes, right? The genotype means constitution of genes. Constitution of genes, right? As for example, here capital T or capital T small or small t, that one is called genotype. Which type of genes are present, right? Alternative genes are present or same genes, identical genes are present. So this is the constitution. Genotype means constitution of genes. Phenotype means external appearance, how they looks. Okay. Sometimes it is also called phenocopy. Phenocopy means two different type of genes. Okay, two different type of plants are. But when we grow them in a same environmental condition, right? Then in a same habitat, so they have a same appearances. Similar appearance between two different type of plants. They are not a single species from a single species, but they are totally different plants. But their appearance is same. So it is called a phenocopy. Right? So phenocopy is another term, and phenotype is another term. Right? Phenotype is physical appearance, how they look, how the plant looks like. It is a tall, it is a dwarf. Dwarfness or tallness, this is a phenotype. Genotype means what? Okay, you can understand when a heterozygous plant is there, that capital T small t is there. In genotype, it is capital T small t. 50% is T, capital T, and 50% it is small t. Right? In genes constitution. So this one called genotype, 50-50%. But phenotype, what is the phenotype? Phenotype means totally and tall. Tallness is the phenotype because this plant is tall. So tallness, it is the phenotype. Okay. Phenocopy means when a pea plant is there, suppose that a pea plant is there, another different plant is there, right? Different plant is there. If their appearance is same due to the habitat, due to the environmental condition, right? Then this one is due to the adaptation. So this one is called phenocopy. Okay. Now come to the some other informations about the Mendel, because he is the father of. Okay, sometimes we use some other words like hybrid vigor or inbreeding depression. Hybrid vigor means what? Bolting effect. What? Hybrid vigors. Hybrid vigor means what? In a hybrid crop, what happened? The production increased. Right? Production increased. This is called hybrid vigor. Right? And inbreeding depression means what? When selfing, through the selfing, right? Then what happens? Their production decreased. Production decreased. That means what? It is called inbreeding depression. Right? So first we come to the Mendel. So about the Mendels in our book is also given something there. Right? Mendel means what? You know Mendel? He belongs to Broome city. Right? The Gregor John Mendel, full name is Gregor John Mendel, and his is born in that 1822 to 1884 he died. That is the date of birth and date of death is there, right? In Silvassa village he born, right? And then after he worked in Augustine monastery. Augustine monastery, he is not a scientist. So because, right, he is born in 22nd, 1822. You can understand 22nd July 1822, right? In a Silesia village, right? Mendel after becoming means when he become adult, then he start work. He get a job in monastery. Which monastery? Augustine monastery, right? As a monk in Broome City, Australia. Worked as monk, right, in Augustine monastery, right? Which one? Which one? Augustine monastery. AGUS, right? Augustine monastery. TARY in Broome City. Okay, in this Broome City, it is located in Australia. Austria. He worked there, and all the experiment which he did, it is in this monastery, right? He started. Second point is here. He started his historical. No, we can say it is historical experiments. Historical experiments from which 1856-57 until up to 1863. On 1856-57 to, right, from 1856 to 57 up to 1863. In this same Augustine monastery, right? Now you understand how many years he spent here. 7 years total, because sometime it is maybe ask how many years he. 7 years, right? And his result, this result means his result, it is published, right, in a science journal. Results are where published in a science journal. What is the name of this book? This journal is Nature for Gender Variant. That name is Nature for Gender Variant. This is the name of the science journal in which his results were published very first in Broome City, right? Because this journal, it is not in English journal, it was in, this is German language is there. German journal is there, German language. The German language, it is, this one is not a German language, but German language means another one is there. It is Varshu, Ober, Flagen, Hybridian, that is different name is there, right? Now here, now next point is there. This journal, who published this journal is published by, it is published by, it is means that science journal, Nature for Gender Variant. It is published by Nature History, right? Nature History Society of Brno, published by NATURE, Nature History, Nature History, right? Society of Brno, Society of Brno. this society, it published this journal in the heading, with the heading, right? Which heading is there? What is the heading of this one? Hybrid, right? Means nature history of Satia Bruno. Hybridization, right? On experiment in plant hybridization. What is the heading of that one? The right. This paper means this one, Nature for Gender Variant. So this paper, right? A paper, we can say, paper named by Mendel, right? Or Mendel named by, a paper of Mendel named by, what is the name of? Experiment in plant hybridization. This is the name, experiment in plant hybridization. Experiment in plant hybridization, right? With this heading, this one is published. But at that time, Charles Darwin is very famous. Charles Darwin is very popular. So the public, he only see the Charles Darwin, and no one see, or no one means that is think about this experiments, because it is, Mendel was not a scientist, right? And at that time, they simply thinks he is a monk. First thing, okay, three other differences is also given, right? First thing is there, communication gap is there. So this one is a science journal, and in this heading, this means experiment in plant hybridization. Simply they see and throw, right? They did not think deeply here. That after what happens? After some time is 1884, Mendel was died, but he did not get any credit about his experiments. He did not get any credit in his life. He did not understand how he becomes a father of genetics. So he did not get any credit about his experiments, right? Mendel died on 1884 without getting any credit for his experiments. Right? He did not get any credit in his life. Right? 1884 he died. Okay. When he died, after 16 years after his death, 16 years after his death, in 1900, three different scientists from different countries, they observed the same thing, right? And Hugo de Vries published this Mendel experiments, means we can here say republished Mendel's experiment, right, in 1901. Then they realized that in 1865, these results were published in Science Journal in which year? In 1865, right? Just 2 years, right, after the completion of this experiment, right? That means they understand that 34 years before, 35 years before, these experiments are also also happened by another. These experiments were also made by another scientist. Scientist not, it is a monk. That is, we can say here Mendel, Georg John Mendel, right? After 16 years off, and this Mendel died because of his kidney. His kidney is damaged. Kidney disease is there, right? So Mendel died because due to the kidney disease, right? And this disease is called Bright disease. Bright disease, name is different. Bright disease, right? So who these three scientists are there who discovered this Mendel's? law. These are Karl Lorenz, Hugo de Vries, right, and Erich von Schermer. After 16 years of his death, it means which? Mendel's death, right? Three scientists, that is means in 1900, three different scientists from different countries rediscovered, right, the same experiments. Same, right? And these three scientists are first Karl Lorenz, C-O-R-R-E-N-S-C, right? Karl Lorenz, C-O-R-R-E-N-S-C. This Karl Lorenz, he belongs to Germany, and his experiments were going on in Mainz, Germany, and he experimented in Mainz, G-Mainz, right? Second one is our Hugo de Vries. Hugo de Vries from Holland, right? And his experiment is on evening primrose. Holland evening primrose plant is. Third one is Yvonne Rich Schermack. Erich von Schermack, right? So Schermack is T-S-C-H-E-R-M-A-K, Erich von Schermack, and he belongs to Austria, and his experiment is not a symbol, but it is, he used different plants. They rediscover the Mendel's law, and Hugo de Vries is created about this. Hugo de Vries, right, republished the Mendel's experiment. This in 1901. Right? They discover in 1900. Right? 1900. In 1900. And 1901 they rediscover this. Okay? That means we can say here, because first publication of first it is published in 1965 and second time it is republished in 1901. That means what? It 34 to 35 years these experiments were hidden, right? Mendel did not get credit in his life. He did not get any enjoyment of his success, right? He did not understand what he discovered. He did not understand.